So I'm Kristen, and I'm in research and development at Castle. And so I thought for this session, or this beginning, we'd just go through just a little review of what tests we offer and what that means. And then, so just a few slides, and then we'll take any questions you have on either the clinical testing or future research for uveal melanoma molecular testing. And so, and I want to thank Melody and everyone else for giving us the opportunity to be at this session and be at the event. It's been really great and great to hear all your questions. And I've learned a lot myself, so hopefully we'll have more good questions here as well. So just to give you a little bit of background about our company, so our Castle Biosciences, we are a laboratory. We do molecular testing for cancer. And the company was started in 2008, and really with a mission to sort of provide molecular testing and testing services for rare cancers as well as underserved patient populations. And so besides we have three tests that relate to uveal melanoma, but then we also have a test for cutaneous melanoma. It's also a gene expression based test like the uveal melanoma.
It's on.
Oh, it's on, okay. And we recently also, within the last year, started doing sequencing, so we'll talk more about that. So we also do that for skin melanoma as well. And then we have additional tests in development. For right now, we're working on one for squamous cell carcinoma, as in addition to the uveal and cutaneous melanoma tests. So our headquarters are in Houston, and then our laboratories in Phoenix, where all the testing is done. This is a picture of our laboratory, so this is where all the testing is performed. So just to review what we're really testing here. So we have, there's two types of genetic material in the cell that we test. And so the first type of material, so we take the uveal melanoma tumor tissue and we take out the genetic material. And so one of those is DNA. And so this is where your genes are. So DNA is sort of segmented into genes, and then like genes that you've heard of today, like BAP1, GNAQ, GNA11. And then these are packaged into chromosomes. So the chromosomes are then in the cell. Sometimes the DNA can become mutated. You heard Dr. Afshar talk about this can either happen, in most cases, sporadically, but then there are some cases where it's a hereditary mutation. And so we can actually read the sequence of the genes like BAP1 and GNAQ and SF3B1, and so report whether the tumor has any of those mutations. And so that's what we have a test for that called DecisionDx-UM-Seq, and we've been offering that for about a year now, and that's just off the tumor, so we don't do any blood testing for that. And then there's another type of genetic material called RNA. So I think Dr. Sato earlier said that the RNA is really the message from the DNA. So the DNA is kind of like the recipe, and then the messenger RNA is made from that. And then the RNA will eventually become the proteins, and that's what carries out the function of the gene. So our other test, which many of you have heard of or had yourself, is our GEP test, is what we call it. It's a gene expression profile test. That's where the GEP comes from. And we measure the levels of the RNA. And so this reflects how active the gene is. So if there's a lot of RNA, the gene is more likely to be turned on and turned up. And if there's low levels of RNA, then the gene is usually repressed or turned off. And so we have two gene expression profile tests. One we've been offering since 2010, and that's the class 1, class 2 GEP test, DecisionDx-UM. And then more recently, in the last couple of years, we've been offering a test for PRAME, which Stefan talked about earlier, and that's called DecisionDx-PRAME. So those are kind of the differences between the two tests. One's more sequencing of mutations, and the other one's looking at levels of how active genes are. And so most often we get a biopsy of the tumor, and so the physician will take the biopsy and then put it into a tube with a little bit of liquid that helps stabilize the sample and freeze that, and that gets sent to our laboratory. We also can test off of, for patients who have an enucleation, we can test off the tumor tissue that's remaining, the block of tissue that's stored in the pathology lab. But primarily we get frozen biopsies. And so, like I said, we use the RNA from that tumor tissue, and that we measure 15 genes, so the levels of how active they are. And then using an algorithm, we can tell whether the tumor is at low risk, class 1A, at intermediate risk, class 1B, or at high risk for spreading, and so within 5 years. And so that's where the class 1 and 2 come from. DecisionDx-PRAME looks at a single gene, PRAME, that we heard about earlier, and that test says whether the tumor has PRAME expression or whether it's negative for PRAME expression. And we see that about 30% of uveal melanoma tumors are PRAME positive, so it's more likely to be negative, but about 30% we see are positive, and that may be associated with an increased risk of disease spread. And then the sequencing test, DecisionDx-UM-Seq, we actually look at mutations in 7 genes. So here we're looking at the sequence of those genes and seeing whether there's alterations in those sequences from a normal gene. So we test for GNAQ, GNA11, CYSLTR2, PLCB4. These 4 genes are fairly common to uveal melanoma, especially GNAQ and GNA11, which you've probably heard of. And then there are 3 other mutations, EIF1AX, SF3B1, and BAP1. And so most tumors will have one of these 4 and then one of these 4, generally. That's not always the case, but usually there'll be one from the group on the top and one from the group on the bottom. And so far it seems like these EIF1AX is associated with a lower risk of metastasis, SF3B1 with sort of an intermediate risk, maybe later metastasis, and then BAP1 is associated with a higher risk. And so these 2 tests are more recent, and this is work that's still being studied in a prospective study that Stefan was talking about, the Collaborative Ocular Oncology Group study. And so they're just, we're trying to offer these tests as more information becomes available. So I probably won't go through all this. I think we get a lot of questions about PRAME and what does PRAME mean. And so I think Stefan covered some of this earlier, but just in case you missed it or want to review it again. So PRAME stands for Preferentially Expressed Antigen in Melanoma. It was originally identified in cutaneous melanoma as being very highly expressed, and it's actually a part of a couple diagnostic tests for cutaneous melanoma. It's not normally expressed in adult tissues, but it does seem to be expressed in some cancers, including uveal melanoma, skin melanoma, some of the myeloid leukemias, and sarcoma. So Dr. Harbour's laboratory found that PRAME seems to be upregulated in some uveal melanoma tumors. We see that in about 30% of the cases. And it seems to be associated with a higher risk of metastasis. And then one thing that's advantageous about PRAME is that it is expressed on the surface of the cell. It's an antigen, so that means that there can be targeted therapies towards PRAME, and so that makes it a good marker. So right now, it seems to have prognostic value, but there are therapies that are being developed and tested that target PRAME, so it could be a therapeutic marker. And so again, this is being investigated with how it works with the gene expression profile test as well as the sequencing mutations to really put all that information together and understand the risk in the best way possible. So I think that's all I was going to really say with slides, but I think I'll sit down and we'll take some questions if people have questions about the test or more specifically about something. Mm-hmm.
My iron was enucleated, but it was about 3 years ago. Do you still get information from that?
Yeah, so so the test is for 5-year risks. So as long as you're, 3 years, it would still be valuable, I think, for you.
It's frozen in UCLA, I'm sure.
Oh, is it frozen or is it, you had the whole eye removed. So yeah, so basically what you would, a physician would need to order the test for you. And so what they'll do is they will fill out a requisition form, tell us where the enucleation specimen is stored, and then our laboratory would reach out to them and coordinate shipping the sample.
So would that be my doctor at UCLA then?
Yeah. And if we can talk a little more specifically too. And
What's the percentage of patients that have, what's the percentage of patients that are 1A, 1B, 2?
So that's a good question. I actually pulled this data recently from a couple weeks ago. So this is looking at, on the left-hand side of all the tumors we've tested since 2010. And so overall, as a population, we see about 2/3 of the patients are class 1, with about 2/3 of the class 1 patients being class 1A, and about 1/3 of those being class 1B, and then 34% class 2. And this is similar to what was found. Dr. Harbour led a very large study, the Collaborative Ocular Oncology Group study number 1, which Stefan talked about earlier, that really validated the GEP test, and they see similar percentages. So it's been fairly consistent over time. And then overall, we see about 30% are PRAME positive. One thing to mention is that one thing we've observed is that class 1A patients are less likely to be PRAME positive than 1B and 2. So if you see in the purple bar, as the class sort of gets increased risk, there does seem to be an increase in PRAME positivity, which I think kind of makes sense. And so that, I think that's pretty consistent with what's been shown research-wise too.
So I asked about PRAME, and I got 50-50. Probably was directed at me as a class 2.
Yeah, I think class 2, you see, this is just what we've seen in about a couple thousand cases overall, but.
Anyone else?
So I was a patient of Dr. Harbour in 2013, and I agreed to the biopsy and to be part of his test. I got the results back as a class 1A, but I never got any results back as a PRAME positive or negative.
Yeah.
Would I be able to find that out? Is that something that was tested back then?
What year about?
2013.
2013. So at
December of 2013.
yeah, so at that point, PRAME, I don't think that they even knew that PRAME was.
Right, yeah, that, you were a little too early, that hadn't, and the issue is that we wouldn't have residual sample retained that far back, so.
I was afraid that would be the answer.
Yeah, we started offering PRAME in October of 2016.
Okay.
You don't need the mic, do you?
No, but, so we did ours in August of this year. And I remember getting your printout back in the mail and, the doctor called us and said, hey, 1A, that's great. We can't remember, my wife is the patient, but between the two of us, we cannot remember anything about PRAME.
About
It was
PRAME.
never mentioned. Will that be on that form? Was that requested also, or did it have to specifically be requested?
So
It's
most,
the number one thing we're checking tomorrow when we go home.
right, and call the lab, yeah, so call us and we can check and see if it was done. And
Okay.
chances are pretty good that it probably was done.
Okay.
It is a separate test,
Uh-huh.
but we don't really require a separate order
Got it.
per se. I mean, we have a way for the office, as they order the GEP, to include PRAME,
Okay.
and we will run PRAME unless they specifically ask us not to. So it sort of depends. I'm not sure who you saw, but if they didn't specifically want us not to run PRAME, then it was likely done.
Okay.
So check with us and we'll be able to give you the information.
Outstanding. Thank you.
Would you still have a sample in case it wasn't done? and they
Yeah,
did
if you're that recent, then we should have residual
And it
sample.
might even be on the printout, we just didn't notice and it wasn't pointed out.
It'll be a separate report.
A separate
Okay,
Yeah.
all right.
It's two separate reports.
I
So.
had to ask my doctor for the PRAME report specifically.
Yeah,
But
okay, good.
I got the one, but didn't get the other.
All right.
Yeah, I think
Thanks.
sometimes, I
Good
mean,
job.
PRAME is relatively new in the hands of the clinicians.
Yeah.
So I think sometimes they get the information and it's, it's interesting to them, but they're not sure what it means or how to discuss it with you,
Right.
the patient, so they may or may not mention it.
Okay,
So just, ask the question and that'll open the way for the conversation.
thanks so much.
Do you have anything to say on PRAME? Positive, negative? Any more questions? You've all had it? you're exhausted, your brain's fried.
Probably.
Is there anything else you want to add?
Nothing specifically. If there's no more questions, I guess.
Yeah, I think the only thing I would just say is like, we're very, you know, as Castle, we're a very small company. And so we certainly would encourage people if you have questions about your, you know, the science, but some of the research or literature on it, or also with insurance and payment of, you know, things like that, please don't hesitate to call us and we'll get you to the right person. So sometimes I think people are hesitant to contact us, but we have our customer service number outside, and we're always happy to help and provide whatever information we can.
So people who want additional DNA testing,
Mm-hmm.
you want to explain that your biopsy is taken and prepared one way and the other is prepped and shipped a different way?
Yes, so for our sequencing testing, we actually so our sequencing testing is relatively new. It's been offered since January, I think, so about a year now. We run it off the same biopsy specimen as the GEP test. So over time, the sort of methodology has improved with the GEP, RNA extraction and things like that, and the chemistry, so we're able to use less sample than we did a few years ago. So we're able to take part of that sample and we can run the DNA testing. So we extract the RNA from one for the GEP and then DNA from the rest of the sample for the mutational sequencing. So for our tests, you can run both from the same biopsy. Maybe I think there are other tests that are run exclusively off DNA. So in that case, our testing, I guess, I don't know if that's maybe what you're asking, but some tests are run exclusively off DNA. So in that case, the RNA would be discarded. And then previously, when we were just using RNA, then the DNA would have been discarded. But right now, we're able to do both from the same sample.
Thank you on that. Anything else for the benefit of all?
You had said that we could call your lab. Would it be easier for those of us that are here to give you our information, and when you go back, could you just do that instead of us going back and calling you and individually thing, maybe have a sign-up sheet for us?
Sure.
Okay.
Yeah, Kristen is our head of clinical operations, so she would be, if you want to give her whatever information.
Well, thank you, doctor. And Castle.